Our first presenter is from Andelyn Biosciences. She is Kristin Heller, Associate Director for Plasmids. Welcome, Kristin.
Thank you very much, and thank you for the opportunity to present. And I'm also very grateful for the opportunity to present on Andelyn's capabilities. All right. As mentioned, my name is Kristin Heller. I am the Associate Director of Plasmid Manufacturing at Andelyn Biosciences, and I've been here for about 7 years. Today, what I'm going to present on, first couple slides are an introduction to Andelyn Biosciences and what we do, and then we're going to switch gears and we're going to talk about considerations when choosing the right plasmid and the right plasmid manufacturer. So an overview on Andelyn Biosciences. We are a CDMO that has spun out of Nationwide Children's Hospital about 4 years ago. Our patients are the focus of everything that we do, and that is what was the inspiration for Andelyn's name. So the A&D in Andelyn actually comes from Andrew, who received the first U.S. investigational gene therapy for Duchenne muscular dystrophy in 2006. And then the ELYN comes from Evelyn, who received experimental gene therapy for spinal muscular atrophy in 2015. So what we are is a concept to commercial gene therapy CDMO. So we are one team and end-to-end partner. So we have different pillars in our organization. So we have the Plasmid organization, which I will talk about here in the next coming slides. We have our process development. So that's process optimization and scale up of viral vector manufacturing. They also do tech transfer of external processes and process characterization. And then we have vector manufacturing. So we have a configurable platform process that spans from research grade through GMP manufacturing. We support adherent systems and suspension with bioreactors up to 2,000 liters. And then we have a fill finish where we can fill various sizes, customizable volumes, and do primary labeling and packaging. Along with that, we have analytical development and quality control, quality assurance, and regulatory support. So we have 3 different facilities located in Columbus, Ohio. So we have the Andelyn Development Center, which is for viral vectors. They support development through tox grade manufacturing. It's a 42,000 square foot facility specializing in suspension and adherent as well. We have our Andelyn Corporate Center, which is viral vector manufacturing. It encompasses tox through commercial AAV manufacturing. It's about 180,000 square feet located on the outskirts of the Ohio State University campus. This is where we have suspension 50 liter up to 2,000 liter and can do adherent platform up to 60 hyperstacks. And then we have the Andelyn Plasmid Center. So this is still located on our legacy facility at Nationwide Children's Hospital. This supports plasmids all the way from research grade through GMP, and it's about 9,500 square feet. So going into considerations when choosing the right plasmid and plasmid manufacturing partner. So what we believe is client delays equal our delays. And so there's advantage of having an integrated approach to both your plasmid and vector manufacturing. We know that access to plasmid DNA is one of the biggest bottlenecks in downstream biopharmaceutical manufacturing. At some point, on average, manufacturers can wait 6 to 10 months to get started, and then upwards of additional 10 months for production, analytical analysis, and release of plasmids, which can cause delays when getting your product to the clinic. We know that beyond manufacturing of plasmids for clinical trials, organizations will spend months to years on the development of plasmids. And so we really believe that as early as possible, clients need to consider the right plasmid and the right plasmid manufacturer to help streamline their development efforts. And that's what we are here to do. We're here to help our clients accelerate their timelines with our plasmid manufacturing capabilities. So the Andelyn Plasmid Center, or what we know it as the APC. So as I mentioned, we have about 5,200 square feet of dedicated GMP space. That includes 5 production suites. And then we have approximately 1,600 square feet of preclinical / process development space. As I mentioned, we do grades of research grade through GMP. We take a single use approach. We offer both shake flasks and fermenter for our clients. And we have a majority of our release assays come from our in-house QC capabilities. So the preclinical plasmids. So we have two different grades here. We have research grade, and this is meant for basic proof of concept studies. So this is mostly, 1 milligram and up. It's very small amounts to kind of get you through that proof of concept to choose the right plasmid for your the rest of your studies. We then have tox grade plasmids. So these are about 10 mgs. We can manufacture about 10 mgs and up. We utilize a single-use product-dedicated materials. This is a very highly documented process. We have product segregation, including dedicated BSCs, incubators. It is very closely to our GMP manufacturing, just in a non-GMP space. And this really helps be able to scale the plasma manufacturing process from your tox grade through your clinical trials. So this plasmid is for IND enabling toxicology studies and can be used for phase 1 clinical trials. This preclinical plasmid space also houses our process development. So that's development of new processes that may come from our clients that involve plasmid DNA or optimization of current processes. One of the big projects we have is, bringing on fermenters and then, any downstream optimization to really increase yields for our clients. We then offer also vector consultation, cloning, and optimization. So we do have our own AAV plasmid backbone that clients can license from us. We also offer DOE optimization and analysis of plasmids for higher productivity. Our capacity, we can do up to 42 liters in shake flasks per day. We can manufacture 4 large-scale productions at one time. We have an Amber 250 system to be able to do DOEs, and then this facility also has a 50-liter fermenter. So going into our GMP plasmid manufacturing, so we do have 5 suites, as I mentioned. Upstream and downstream are done in one room. So when your plasmid goes into that room, it does not come out until the product is filled. We have rooms to accommodate shake flasks or fermenters. A lot of our clients are talking about, treating ultra rare diseases. Some of our clients need, very small amounts of plasmid, and then some need, very large amounts of plasmid that are trying to treat systematic disorder. So we really want to be able to cater to our clients at any stage and basically not eliminate somebody from coming to us that may have something that is, small. And like I said, we want to be able to be there for our clients at any stage. Our GMP capacity is up to 54 liters in shake flasks per day, and then we are bringing on a 50-liter fermenter in early 2025. So this is the analytical capabilities for plasmids. So as you can see, we have our 3 different grades of plasmid, and you can see that as we go from research grade upwards of up to GMP, you can see that the testing panel increases. I will also like to mention that almost all, so all but 3 of our analytical assays are done in-house. And this, we really believe, also helps accelerate our clients to get to the clinic as fast as possible. We're not waiting for having to send out, plasmid for testing to outside facilities and be at the mercy of their timelines. So a little bit more about our different capabilities. So we can do cloning, as I mentioned. We have yield assessments and DOEs for all stages. We obviously generate C of A's. We can do E. coli master cell banks. There's plasmid stability that we can perform for our clients on their plasmids. As I mentioned, the big thing on our tox and GMP is using single-use or product-dedicated materials that help prevent the chance of cross-contamination. We can offer TSE, BSC statements. The GMP, as mentioned, is manufactured in a clean room and has QA oversight. So additional offerings, as I mentioned, we can do master cell bank generation for both research grade and GMP. We can perform stability on master cell banks and plasmid products. As I mentioned, we have an integrated offering of AAV and plasmids. This gives our clients one single CDMO to manage under one MSA. The nice part too for our company is that there's an integrated project manager. So if you come in and you get one project manager from basically plasmids through process development, through preclinical, all the way up through commercial manufacturing. So we get to have a really good relationship with our clients and really be there for them as they navigate their way through process development through the clinic. And we have one quality system. And then the AAV and the Plasmids teams, we actually exchange technical information quite frequently. There's many times that I'm on a call with the process development team, and we're discussing what are the plasmid needs, how do we change if those plasmid needs changes. We really are very closely associated with each other. We also can leverage a plasmid DMF, as I mentioned, in-house plasmid QC capabilities. We have make-to-stock helper plasmids. So we have 2 ad helper plasmids that are available for our clients to use. We manufacture them to be off the shelf. So when a client comes to us, depending on what plasmids they want to use, we can have a capsid. So there's a couple of capsid plasmids that are available along with ad helper plasmids, so that then our clients only have to worry about manufacturing their gene of interest. And this also is meant to accelerate our clients as quickly as possible to the clinic. They are available free to use for research applications. They have optimized functional sequences, and they come in kanamycin resistance. So finding the right plasmid, some considerations to think about. So choosing the right plasmid, we've seen this for our clients before. Choosing the right plasmid upfront can help prevent costly delays in the development life cycle. So things to consider: antibiotic resistance. So we always talk to our clients upfront and try to find out where they're going to do their clinical trial, because where they do their clinical trial is going to depend what antibiotic resistance they can use. So we've had clients come in with an ampicillin-based plasmid. We ask them, are you planning on doing a clinical trial in the EU? They say, yes, we plan on it. And then we have a discussion that says, you might need to change your antibiotic resistance. We're available to do that for them as well. Looking at ITR integrity, making sure there's intact ITRs present. These are very unstable regions. Bacteria don't really care to replicate them while dividing. And so there can be deletions. And we really take care when we're looking and screening to maintain ITR integrity. Discussion on transgene cassette size. AAV only packages up to 4.7 kb. Most genes are a lot larger. And just making sure clients are coming in with the right size cassette and helping prevent any potential issues they may see in the future. And then another big thing, our AAV backbone has a stuffer sequence in it to help prevent reverse packaging of the backbone. So sometimes there's very small backbones. And so AAV can potentially package those bacterial backbone, and that's not good. So we've made a backbone that's large enough that won't get packaged into AAV. And then there's some things to consider depending on the client, whether or not you include regulatory elements like WPRE sites. This is not limited to vector plasmids. There's also the same considerations that come for your capsids and your ad helper plasmids. Our capsid plasmid is modified to potentially prevent recombinant AAV from being made. And really choosing the right plasmid grade for your needs. We do offer 3 different grades. Our tox grade we've seen before, where there's been plasmid contamination from a plasmid manufacturer and has caused delays and issues in their toxicology study. So we really take a high level of quality to our tox grade plasmids and to really make sure that we're setting our clients up for success. So partnering with a supplier that can scale alongside your therapies development can save time and money. So we do have standardized connected protocols. So your protocols from process development through GMP are very similar. We have comprehensive data collection. So we're constantly collecting data every time we manufacture the plasmid or vector. And so that we can be able to take a look and see how is it performing? Is this run the same as the last run? Is it worse? What happened? And really be able to do that analysis and truly understand how this is manufacturing. We do have in-house expertise. We have access to changing regulatory environment. We're very close with our clients. They're providing feedback, the feedback they're getting from regulatory agencies, and we can kind of, help guide them, and they can also help guide us into making sure that we're really setting everybody up for success. As I mentioned, vertical integration from start to finish. You never have to, it's really great to never have to leave your CDMO. We can leverage a DMF for access to intellectual property that will help our clients with speed and efficiency when filing INDs or BLAs with the FDA. And then just, strategic partnerships. One of our minority investors is Danaher. So we do have access to the Danaher portfolio of equipment and expertise as well to be able, again, to help support our clients. We believe vertical integration is key. As I mentioned, that's why we are a full service CDMO. So each piece of our manufacturing team works closely basically from start to finish to get your product to the finish line. As I mentioned, personalized business experience, you get to know your project manager very well over the many years that you are working with them. And then we have a strong supply chain relationship. So we have a unified supply chain where it's much easier to manage delays when everything is in-house. So if there's something that's delaying plasmid manufacturing and you were supposed to go on a slot for AV in a month coming up and we know it's not going to be released in time, we can shift the schedule. We know where it's at. We can plan for that and get you in the next available slot. When you're trying to manage from another CDMO, you're really at the mercy of when they can get it to you, and then when the next CDMO is available to put you in that queue. And we really try to work with our clients to make sure that if we have a delay, it's not delaying them further. And that is the end of my presentation. Again, I want to say thank you for the opportunity to present, and I'm happy to answer any questions you may have.
Thanks, Kristin. Great presentation. A couple things I want to ask about. I really love your one slide about considerations and finding the right plasmid and considerations for that. When do you recommend that a drug sponsor, a client, talk to you about that or reach out to you for that outcome? And how long does it take? That second part's probably, well, it depends, but I'll ask it anyway. Yeah.
Yeah. So, we recommend as early as possible. We start talking to clients before they even have an idea of what they want to do. They're like, I think I want to treat this and I don't really know, and I'm very new to the field. Can you help us? I think those are the best options, because the earlier you can get to it, the better and more successful you'll be, and the less money and time you'll waste. And yeah, it really depends on the plasmid. We see differences, in self-complementary vectors versus, single-stranded. There's so many different considerations of, short hairpins and RNAs, and it really just depends. Unfortunately, I don't have a magic answer for you. It depends. We're pretty quick. A lot of our optimization, looking at that is a few months. So it can be very, very fast.
I suspect there are a lot of people that you described that are like what you described. There's so many innovators in this space that I'm glad you mentioned that, because I think we all want that to happen quickly. Right. The entire industry and society needs that to happen.
Right. And
I was going to say, go right ahead.
I was going to say, and you figure most of these are, family foundations, parent foundations that don't have a lot of money to spend. So, you want to utilize that money that they have to the best of your ability for them. So
that's a great point. I'm glad you made that. Yeah. Something else you said, I may not get it right. The plasmid, it's the one room concept. The plasmid comes in upstream, downstream. It doesn't leave that room. Yep. How important is that, and what benefits are delivered because of that model you have?
I think for us, I think it goes quicker. So you're really, a lot of the delays will come from like room changeovers, cleanings, getting ready from the next product. And so if you're staying in that same room, you don't have to wait for the next room to open up to get into purification. It's already there, and it already just runs through. So that's why I think the advantage of that is that, you're there and it's your room for however many weeks it takes us to manufacture, and then you're good to go.
Great. And then just finally, how much capacity do you have? How many are you ready to, is there any wait for all of the things specifically that you spoke about under your purview in plasmids?
I was going to say, no, we definitely have capacity, and I would recommend reaching out to our business development team and have a conversation with them, and we can definitely help our clients as soon as possible.
Great. Well, thank you, Kristin. Great presentation. I love your founding story. I really, it kind of reminds us all why we're all here. So thank you for doing that every time you present.
Yep, you're welcome.
Kristin Heller from Andelyn Biosciences. Thank you very much for being with us today.